In this video, we are going to be looking at an introduction of DNA technologies and how we make recombinant DNA. The first topic that we are going to discuss is restriction endonucleases. These are also known as restriction enzymes. These restriction enzymes, understand that are going to be hydrolases because they are going to cut the backbone of DNA molecules. So as you can see, restriction endonucleases digest double-stranded DNA at specific sites. Understand that they act, if we want to think of an analogy, because they cut the backbone of DNA molecules, they're going to be acting as scissors of DNA sequences. And they're going to be cutting DNA into single genes of interest. So it's going to be cutting them into smaller pieces. When it comes to these specific sites that they identified, understand that the recognition sites for restriction enzymes are going to be palindromic. What that means is that if you read them in any direction, so to give you guys a word example, would be like the word aviva, which means that if you read it in the forward direction from left to right, you have an A, V, I, V, A. If you read this word backwards, meaning reading right to left, you still have A, V, I, V, A. So when it comes to this, understand that these recognition sites have different sizes and they have unique sites. And the cutting specifically that these restriction enzymes are going to do will be between the same nucleotides on each strand. So to give you guys examples of restriction endonucleases, here we have the two different types of cuts that they can perform on a double-stranded DNA. The top example is what we called in molecular biology sticky ends. And the reason why we call it sticky ends, as you can see, is because it's going to have protruding ends on either side that it is cut. For example, when we look at the enzyme PST1 and it is digested double-stranded DNA, we can read this strand in the 5' to 3' direction, in which now we have C, T, G, C, A, G. And as you can see, if we look at the complementary strand and we read it 5 to 3, it's going to be C, T, G, C, A, G. Regardless of any of the two strands, specifically PST1 is going to cut at those sequences in between the A and the G. Remember, the cutting is happening at the backbone. What we are hydrolyzing is the 3' to 5' phosphodiester bond for those sequences of DNA. We also have blunt end cutting, which is what is illustrated in the bottom example. And in blunt end cutting, as you can see, it's going to be producing two pieces of DNA that is going to have blunt ends. So as you can see, they're going to be flat. Now, to give you guys an example of a blunt end cutting enzyme, here we have EcoR5. And when EcoR5 digests DNA, the sequence, if we read it in the 5' to 3' direction, is going to be GATATC. And then again, the complementary strand, if we read it in the 5' to 3' direction, is going to be the same. So when the cutting happens on both strands, as you can see, it's going to be happening in between the T and the A, and what we produce is going to be blunt ends. Understand that many we don't have a specific number. There's a variety of restriction enzymes, but many restriction endonucleases are going to be producing specifically sticky ends. It depends on what your gene of interest has incorporated in its genome, meaning in the sequences of DNA, that you're going to be using a particular endonuclease in order to cut it. But understand that many restriction enzymes are going to be producing sticky ends, okay? And it is produced because of the specific way that these restriction enzymes are going to be cleaving those target strands. Understand that this is just another example of a sticky end. So for example, the enzyme EcoR1 specifically is going to find a sequence that is GAATTC, and right there is going to do a sticky end cut in between the G and the A in either direction. Now, if there's no ligase, meaning no enzyme that links sequences, these ends are going to remain separated, and because of hydrogen bonding events, the sticky ends are held, are going to be held as a structure. Okay. Understand that the reason why we need to talk about restriction enzymes is because they are involved in the process of creating recombinant DNA. By definition, recombinant DNA is going to be DNA molecules that contain covalently linked segments derived from two or more DNA sources. When we are combining these two or more DNA sources, okay, and we make a construct, that's what cloning is defined as. Now, understand that the combination of specifically a plasmid vector and foreign DNA is what creates a chimeric plasmid. Okay, we already defined in previous chapters what plasmid vectors are, and remember that a plasmid is going to be the vehicle that it is utilized to produce DNA or protein, depending on what you need to utilize it for. The foreign DNA that is present is going to be the DNA of interest, meaning what you are trying to study, okay, in the context of the cell. So understand that when we are trying to synthesize these systems, we're going to have restriction endonucleases or restriction enzymes are going to be cutting the segments of DNA in both the organism that is going to be the DNA of interest. And as you can see, in this example, the DNA organism 2, let's assume that that is going to be the plasmid vector. Then ligation, so in the arrow, that's when we have a ligase putting the sticky ends together, and what results when we unified a plasmid vector with foreign DNA is what we know as a chimeric plasmid. This chimeric plasmid can be utilized in other experiments in order to produce more DNA of the DNA of interest, or if you're interested in protein studies, you can put it in a specific bacterium, similar to what we discussed previously in Chapter 5. You can produce then the protein of the DNA that you're interested in and do research on it. So, understand that plasmid is going to be a circular piece of DNA. Many plasmids naturally occur in bacteria, and understand that bacteria actually naturally trade them around each other. Now, Plasmids that we utilize in cloning are utilized in genetic engineering for expressing DNA or expressing protein. The main parts of a plasmid are the following. So, a plasmid is going to have a resistant gene. As you can see in this case, it's just saying that it is for ampicillin. Ampicillin is going to be a type of antibiotic. And the reason why we need that is for selection of the organism that has the chimeric plasmid of interest. Plasmids also include a multiple cloning site. And this multiple cloning site is what contains the restriction sites or restriction enzymes where the DNA must be cut. Most of the time in this area of the multiple cloning site is where we incorporate the DNA of interest. And understand that typically when we are trying to incorporate our DNA of interest, we use at the 5' end and at the 3' end different restriction sites. So there's going to be, in the case that we utilize sticky ends, there's going to be different sequences in which we know that our DNA is going to be sitting in. Because if you use the same one, you may have problems in the future when you're trying to do cloning. Now, understand that in this figure, they also express that you have the useful gene we want to insert into the bacterium. But as I express, most of the time, the gene that you're interested in or the DNA of interest is going to be cloned into the multiple cloning site. Okay? Understand that there's other components of the plasmid that are not illustrated in here. And some of them we already seen in other chapters. So other components for plasmids include a promoter region. And this promoter region is what is going to be important in transcription in order to create the mRNA that then will be translated into protein. We're going to have an origin of replication. And this origin of replication site is where we are going to be making more DNA in the case that we need to do that. And as you can see, so the 4 essential sites, just to number them, is going to be the resistance gene, multiple cloning site, promoter region, origin of replication region. Okay? And the 5th one, in a way, is going to be the gene of interest, but the plasmid itself, when it comes at first, has these 4 features. Okay? So, how do we make a chimeric plasmid? Understand that there are going to be some restriction enzymes that are going to be cutting the DNA, and specifically because we want to be cutting and pasting our gene of interest, those specific restriction enzymes you're going to be selecting from the multiple cloning site. And as you can see here, this is illustrating the cut and pasting of a gene of interest where we use sticky ends. And in the system where you utilize sticky ends, just imagine that this is kind of like creating ends that is like Velcro, in which now your gene of interest is going to match a corner of your plasmid, and that's why they're able to come together.